Now we are going to see the new practical, that is the double standard permanent micro preparation. Here the meaning of the experiment shows double means by using the two stains. Micro, that is a slide preparation. Permanent, the slide is prepared permanent, and when we get in future time, then we will study the slide. Therefore, this in title of the practical is the double standard permanent micro preparation. Here we are going to see the tissue which are present in the plant body. For this purpose, we have to take the TS, LS, RLS of the given plant material and to see the difference in the tissues. For the difference in tissue, generally they are attached to each other, and only by using the one type of the tissue it is impossible. Therefore, two stains are used. Here, safranin is used. This safranin is used the tissues present in the plant body, which are the dead tissues. The xylem and sclerenchyma are the lignified dead tissues are there, and after staining of this safranin, they become faint red in color. Here another stain is used, light green. Light green, that is, it is known as a faint light green. The tissues, remaining tissues, they are after staining they becomes a light green, means contrast color, dark color safranin and faint color light green, and therefore the whole tissues are stained. And on the basis we can separate the tissue from each other. On the basis the dark colored structures which are present there are dead tissues, and faint light green color which are present, they are the living one. They are used with the help of the light green. So the plant material which are given, the thin sections are taken 10 to 15 out of the floating small sections which are present. The sections are selected. This is the procedure of the double staining. Select the thin sections. Section should be entire, section should be plain, and section should be thin. The selected sections, 2 to 3 sections in a cavity block which contains the chemicals. Through this they are transferred. Here the process, 30% alcohol, 30% alcoholic safranin. Each and every year the percentage which are given, the alcohol is used. Here the steps are upgrading, means as in the tissue water which is present, to replace the water, the alcohol grades are given. If the water remains in the plant tissue, the tissue becomes decayed. Therefore, the 30% alcohol for 5 minutes, 30% alcoholic safranin for 15 minutes. After staining which are given, they are washed with the distilled water, and then 50% alcohol for 5 minutes, 70%, 90% for 5 minutes. 90% alcoholic light green, means stains which are given in 30% and 90% alcoholic light green given. After staining, the section should be washed with the water, then in absolute alcohol 100%. Then alcohol and xylene percentage is 3:1, 1:1, 1:3 for 2 to 2 minutes, and in a pure xylene the section passes. In the pure xylene, the whatever impurities are present and the alcohol which is present, the alcohol is completely replaced by the xylene and it clears the section. And this section is bound in Canada balsam and Depex. Depex or Canada balsam, these are the mounting mediums, and its refractive index is similar to the glass, near about the 1, and they are the clear, transparent, having the adhesive, and it does not decay, and observed under the microscope. The plant material which are given, they are observed under microscope, and tissues are studied.